So first off, I want you to completely cross off objective 4. So we're not going to speak about the regulation of calcium or calcium balance at all as it pertains to the kidney. So you can completely cross that objective off. We're going to look here at sodium balance and potassium balance. So our first objective is to describe how aldosterone and atrial natriuretic peptide regulate plasma sodium levels, and then include an explanation of the stimuli for the release of these hormones, including that of, the release of these hormones. So how these hormones are released, and then how they influence sodium reabsorption. And then the second objective was to describe the major mechanisms whereby water-sodium balance influence mean arterial pressure. So this is a really important relationship for us to start off with. Sodium, water, and mean arterial pressure, okay? The product of our blood pressure is sodium concentration and water concentration. And I talked about that in our previous slide, in our previous packet. Sodium is going to influence water movement. In other words, water is going to follow sodium. If we have excess sodium in our plasma, we have excess water in our plasma, we have excess mean arterial pressure or a high blood pressure. If we have too little sodium in our plasma, we're going to have too little water in our plasma, we're going to have an inadequate or decreased mean arterial pressure or decreased blood pressure. Okay, so these three things are extensively connected: sodium, water, and mean arterial pressure. Okay, and so we can speak about hypernatremia, which is a state of high plasma sodium concentration or high blood pressure. We can speak about hyponatremia, a state of low plasma sodium concentration or low blood pressure. And so we know that sodium needs to be at a very regulated level, a regulated plasma concentration, in order to maintain a regulated blood pressure. This is why if you have hypertension or if you have high blood pressure, the first thing your physician asks you to do is decrease the amount of sodium in your diet. Excess sodium in the plasma is going to further increase your blood pressure by further increasing the osmotic ability, right, pulling more water into your plasma. So sodium and water tie together with blood pressure. We know that sodium is important for two things. One, the osmotic pressure of our plasma, basically our blood pressure. And two, the function of our excitable cells. So when we talk about excitable cells here, we're referring to muscle cells, we're referring to neurons, and we know that their mode of communication, their functioning is heavily reliant on sodium, right? Think about an action potential. An action potential is nothing but opening up of voltage-gated sodium channels, sodium rushing in, depolarizing that membrane potential, and firing an action potential via the movement of sodium, specifically the depolarizing event of that action potential. So these are the two important reasons that we need sodium in our ECF, and we need to keep it regulated in our ECF. So sodium is freely filtered across the glomerulus, right? It's an ion, it moves across without any impedance. It's reabsorbed in the proximal tubule, the distal tubule, and the collecting duct. About 70% of the sodium is reabsorbed. About 30% does get excreted in the urine. It is not secreted, so only what is reabsorbed is eventually only what is filtered, that 30% is eventually excreted, and the remaining 70% is moved back into the plasma, is reabsorbed. The regulated reabsorption, and when I say regulated here, I mean the reabsorption that takes place in the distal tubule and the collecting duct is under the influence of aldosterone and ANP. ANP, or atrial natriuretic peptide, is a hormone that's secreted by the heart, and we'll talk about that later on, but these two hormones are what regulate the amount of sodium that is reabsorbed, right? Sodium is not secreted. Across the proximal tubule, we know that this is not regulated. It just happens freely according to the concentrations there. We know that this reabsorption takes place via active mechanism, so that means it's going to require ATP. And so this happens with the use of the sodium-potassium ATPase pump that is located on the basolateral surface of these principal cells, right? And so let's look a little bit closer here at one of these principal cells. So this is a principal cell. I'm sorry, no, this is a proximal convoluted tubule cell. And this is how sodium is reabsorbed here, again, unregulated, and it's moved passively across the apical surface, and then it's pumped actively across the basolateral surface. It's moved either via co-transport or counter-transport, meaning that it's going to happen passively. And then according to the sodium-potassium pump, it's going to be pumped across into the plasma. In the distal tubule, this is the principal cell. So this is how it's absorbed or reabsorbed across the principal cell. Again, it's going to be moved passively into the cell itself, and then it's going to be actively pumped across the basolateral membrane, right? I do want to point out here one difference. Oh, no, sorry. Okay, so yeah, so this slide is showing that. So this slide is showing the sodium-potassium pump that's being used to actively facilitate the absorption here across the principal cell. And just to distinguish here, I'm using the word principal cell. I don't remember us kind of distinguishing that, but the principal cells are the cells of the distal tubule and the collecting ducts, right? The principal cells are the cells that we call these cells that are in the distal part of the tubule and along the collecting duct. So these are the epithelial cells that are uniquely named in this part of the tubule. So let's move on here to look at the hormones that influence sodium reabsorption. The first hormone is aldosterone. Aldosterone is secreted from the adrenal cortex. It's going to act on the principal cells of these late parts of the tubule. That's the distal tubule and the collecting duct. And it's going to stimulate these cells to do two things. One, increase the number of sodium-potassium pumps on the basolateral surface. That is going to facilitate more sodium being reabsorbed actively into the plasma. The second thing aldosterone does is increase the number of sodium and potassium leak channels that are on the apical surface of these principal cells. Okay, and so those two events, more leak channels on the apical surface and more sodium-potassium pumps on the basolateral surface, together facilitate the movement of sodium from the tubular fluid back into the plasma. Alrighty. And as you can see here, aldosterone is a hormone. It's coming from the blood and it's binding to one of these receptors and triggering these two events. Let's talk about how aldosterone is actually released from the adrenal cortex. So aldosterone is secreted from the adrenal cortex under the influence of the renin-angiotensin-aldosterone system. This is sometimes referred to as the RAS system, R-A-A-S. And this is basically a sequence of events or a sequence of signals that are triggered from the kidneys to then tell the adrenals to secrete this aldosterone into the blood. So the granular cells of the juxtaglomerular apparatus, if you remember in one of our previous packets, I talked about the juxtaglomerular apparatus or juxtaglomerular cells in the kidneys, a cluster of three different types of cells. One of those types of cells were called granular cells. And the granular cells secrete a substance called renin. Now, when renin enters the blood, it is then converted to angiotensin 1, okay? And then angiotensin 1 is converted via another enzyme called ACE. ACE stands for angiotensin converting enzyme. This is located in the walls of our capillaries, in our capillary endothelial cells, specifically in the lungs. And this ACE enzyme is going to convert angiotensin 1 into angiotensin 2. Angiotensin 2 is then going to trigger the adrenal cortex to secrete aldosterone. So as we can see here, this is like a chain or a sequence of events. Renin triggering angiotensin 1, triggering angiotensin 2, which triggers aldosterone. Okay, now let's look a little bit closely at this, this schema of events here. So angiotensinogen is actually always present in the plasma. It's secreted by the liver. And as we can tell from the suffix gen, this is an inactivated version of this angiotensin. And it's always in the plasma, but when it comes in contact with renin, secreted by the granular cells of the juxtaglomerular apparatus, those two are then converted into angiotensin 1. Angiotensin 1, under the influence of ACE, which is angiotensin converting enzyme, will then be converted to angiotensin 2, and then angiotensin 2 is going to be what stimulates the adrenal cortex to secrete aldosterone. Okay, so kind of one thing triggering the other, and then we get the cortex of the adrenal releasing that aldosterone. All right. Now, angiotensin 2, I cannot speak today. Angiotensin 2 does a few other things other than secreting aldosterone from the cortex. It's going to signal vasoconstriction of the sympathetic arterioles. That is going to help increase mean arterial pressure. It is going to influence the posterior pituitary to secrete ADH. That's going to facilitate increased water reabsorption via the aquaporin 2 mechanism. That is also going to increase our plasma volume, which will increase mean arterial pressure. It's going to increase the hypothalamic neurons, which are basically triggering increased thirst, right? Our thirst centers in the brain. So we're going to drink more fluid under that influence. That is also going to increase our plasma volume, which will then increase our mean arterial pressure, right? And then it's going to increase the adrenal cortex to secrete aldosterone. That's going to facilitate more sodium reabsorption via those two mechanisms I talked about, which are increasing the sodium-potassium pumps on the basolateral surface of the principal cells, increasing the sodium leak channels on the apical surface of those principal cells. That'll facilitate more sodium reabsorption into the plasma, which will increase the osmolarity of our plasma, and that will also trigger increased ADH secretion. If we've got a high plasma osmolarity, we're going to increase ADH. Remember, that's one of the two stimuli for ADH secretion that we talked about just now on that checkpoint question. Increased plasma osmolarity and decreased bioreceptor stretch. So that's going to trigger increased ADH secretion, which will then further trigger that rise in plasma volume and that rise in mean arterial pressure, okay? And this is tying back to that connection that I talked about earlier. Increased sodium, increased fluid, increased pressure, okay? And we're gonna see that the opposite is true. Decreased sodium, decreased fluid, decreased pressure. Let's talk about some of the stimuli for renin release. So there are three main stimuli for renin release. One is the decreased pressure in the afferent arteriole. So the amount of pressure that we see in the afferent arteriole that's coming into the glomerulus, if we decrease that pressure, that's going to be a trigger or a stimulus to secrete renin. The second is an increase in the sympathetic activity, right? If we decrease our mean arterial pressure, we're going to increase the sympathetic nerve activity. That's going to increase our renin release, right? And then the third trigger is decreased sodium and chloride concentration in the distal tubule filtrate, right? The presence of sodium or the concentration of sodium and chloride in the filtrate that's coming through the distal part of the tubule will then decrease. If we have a decrease in that sodium concentration, we're going to increase our renin release. And if you think about the job of renin, remember renin is eventually going to trigger aldosterone through that renin, angiotensin, aldosterone system. So the triggers for secretion of renin are going to be decrease in plasma concentration of sodium or decrease in blood pressure. And these three triggers will be in alignment with that type of event. And so if we increase renin release as a response to these three triggers, we will then increase our blood pressure and increase our GFR, okay? And I hope that that was a bit clear, but we'll talk a little bit more about that using this flow chart. So here we see that we drop mean arterial pressure. And so if we drop mean arterial pressure, we're going to need to increase aldosterone in order to fix this mean arterial pressure decrease or decline. And so we're going to increase renin, and the three things that trigger this renin release are a decrease in afferent arterial pressure, an increase in our sympathetic activity, and a decrease in our sodium and chloride concentration in the filtrate in the distal tubule. And so when we have these three events, we will then increase our renin release, which will eventually bring back up our blood pressure, because we can then secrete aldosterone and reabsorb more sodium and